Okay, according to my watch, it's noon. That clock in the back seems to be slow, but we'll go by my watch. Welcome to BioSci 121, Immunology. I'm Professor David Fruman. My co-instructor will be Professor Craig Walsh. And I'd like to start by going through the syllabus and talking a little bit about enrollment. Then there'll be an announcement from a tutor, and then we'll jump right into the lecture. So I projected the syllabus up here. We actually have 3 teaching assistants. We've added a third one, Warren Playstead, but you won't be interacting with them because there's no discussion section. The teaching assistants are really just here to proctor exams and to do grading. And so that brings me that noise to another announcement which I always make, which is please turn off all electronic devices or silence them during the lectures. And if you're going to use your computer, please use it for course materials and not for other things that may distract those around you. Okay, so the textbook shown here, we were a little late in ordering, but I think it should be in the bookstore by now. But it also comes as an ebook, which is what I use. And it has all the information that you need there. And you can, don't have to carry around the textbook everywhere. And so that's a choice for you. And then what I want to talk about next is the examinations. There are only 2 ways to earn or lose points in this course. It's just the midterm and the final exam. So you got to be ready for those. The midterm is 100 points. The final is 200 points. The midterm is a 50-minute exam, and the final is a 2-hour exam. And I've scheduled the midterm this year to be about 1/3 of the way through the course. The final will cover mainly the last 2/3. We consider it comprehensive because the information you learn in the first third is important to understand to apply to the second half, but we won't ask you detailed questions about the first part. So it's a little bit different than some other core classes where what you learn before the first midterm or before the second midterm is really separate from what you learn later on. It's a comprehensive exam, yet it really focuses on the material after the midterm. Our grading system is a little unusual in that we don't use the mean and the standard deviation. We basically use the median, and we break down into the grades into 8 divisions. So 1/8 or 12.5% of the class will get an A, 12.5 A minus, so 12.5 each of the half grades down to a C, and then anything below a C, C minus, D, and F will represent the last 12.5%. So when you look at the midterm and the final, you'll be looking at the median and seeing where you are relative to that and relative to the graphs that we show. The standard deviation is not taken into account. And what this means is that half the class will get a B or better, half will get a B minus or below, and so it's a little more generous than in many of the core classes where they set the mean at the border between the B minus and the C plus. Now, if everybody does really well, spectacularly well, we don't have to give so many grades, low grades, but in general, things sort themselves out, so this is the breakdown that we get. All right, so be aware that the midterm takes place in class on October 21st. The final will be at a different time, 1:30 to 3:30 p.m. I'm going to have, at least for my part of this course, some optional quizzes that I'll post on EEE that you can take, and then I'll post the answers about a week later so you can check your work, so to keep up and see how you're doing and see the format of questions that will appear on the exams later on. I will have a review session. We will have one along with Dr. Walsh on the Friday before the final exam. If there's time, I'll answer a few questions before the midterm as well. I also take questions during the lectures, and I can usually stick around for about 5 or 10 minutes after the lectures to answer questions. But other than those times, the time to meet with me would be during my office hour, which is Monday. I've set it from 2:30 to 3:30 p.m. so that people who have 2 o'clock classes or 3 o'clock classes will still have a chance to squeeze in some time. If you can't if every week you're booked for this entire time and you need to schedule time with me, send me an email and we'll see if we can work something out. But I would prefer to have you prepare your questions and meet with me after class. I have a lot of other teaching duties and research and administrative responsibilities. So I also will have a message board where you can post questions that I'll try to answer that may be frequently asked by other students as well, and sometimes other students will try to answer the question on the message board, and we can deal with difficult problems that way as well, or commonly occurring questions. Now, I just want to go through the syllabus the lectures itself. You can see that I've listed the titles of the lectures, the textbook chapter that they're associated with, and the lecturer over here. And for the most part, we go in order. And the first half or so of the course deals with basic mechanisms of the immune system, the cells and the molecules that do the work in the immune system, how they're put together, their general functions. And then the second half of the course really puts this in motion and applies it to problems of host defense to infection, autoimmune disease, and transplantation, and so forth. So the first half is really mechanistic. You might think of it as a fair amount of memorization of parts that are going to later be put together into some sort of whole. And sometimes students complain that there's really not enough context in the first half of the course. So what I've done this year is I've moved up a lecture on the function of antibodies from chapter 9 to follow the lectures on antibody structure and generation of diverse antibodies. And when I looked at this lecture and the content, I realized that it really doesn't require any of the material in between chapters 5 to 8. So it's possible to give this lecture up here, and all the burning questions you have when you're learning about antibody structure and diversity will start to be answered and even before the midterm. So you'll get a little bit of application in the first half of the course. The only other point I want to make is that the other part of chapter 9, I'll be giving that lecture on November 1st, which is before Dr. Walsh will present chapter 8. And again, the B-cell immunity lecture does not require prior knowledge of the material in this chapter 8. And this is done based on the schedules of myself and Professor Walsh. Okay, any questions about the syllabus so far? I want to go back for a second and talk about the book. You don't absolutely have to buy the book or read the book. I recommend it, but the first and most important thing that you should do to prepare for exams in this course is to really understand the lecture notes. Myself and Professor Walsh both provide notes and slides. And we do not ask you any questions based on material that's not presented in class and summarized in these lecture notes. Our lecture notes are fairly detailed, so if you read and understand everything in these lecture notes, that is sufficient. Now, the reason for the book is that often the material is not exactly clear, or you want to put it in the context of some of the human diseases that they talk about in the textbook, or restate the information. And probably the other important reason to get the book is because the pictures I'm going to show you, the slides I'm going to project, will not have the figure legends or captions with them. So sometimes when you go back and look at the slides during your review, you won't really remember exactly what it was trying to say, even if you can refer to the notes. So if you actually have the book or the ebook, you could look at the captions with the figures and it'll remind you exactly what's there. And the other really important reason to get the book is that the beginnings and ends of the chapters have summaries that really put everything, big picture in context, and the important facts and concepts for you to remember. And we really try to be concept driven in this class. There is a lot to memorize, but if you don't understand the big picture of how cells work together and how the immune response happens and what goes wrong in different diseases, then you're not going to do well on the exams, especially the short answer, free answer parts. So by reading those more broad summary material and also looking at the sample questions in the back, you put yourself in a better position to do well. But just to return to the first point, the very first thing and the second thing you should do is review the notes, because we will choose our questions based on what we presented in class, and we will never, I promise you, have a question based on material that's in the book that's not covered in class. So if you're reading in the book and there's something that's unfamiliar and you don't understand it, don't worry about it if it was not in class. Don't spend your time, your valuable study time, trying to understand everything in the book if it wasn't presented here. And every year we have people who come after midterm and say, I did really badly. I read the book and I did badly. And I'll say, well, how well did you study the notes? And they'll say, well, I looked at them, but I really read the book. And for some reason, the message doesn't always get through. But focus your study time on the notes and use the book as a secondary study tool. Okay, now everybody who's going to be here is probably here. I can talk about enrollment. You can see this room has extra space, but we limit the class to 160. Some of you may be here waiting to get into the class. I can assure you that you will get into the class because I've been teaching this class for 12 years now, and we've never had an undergraduate student who hasn't got into the course. And that's because within the first 2 weeks, we have a large turnover. A lot of students will drop the course, will choose something else. They find my lectures boring or challenging. I don't know what it is, but I scare them away. So you will get into this class. The reason we don't open it up to 250 or whatever the capacity is, we used to be in a smaller room. This class is designed to be a reasonable size. We have a limited number of TAs, and it used to be 135. We only raised it to 160 this year because we got a third TA. It's just impossible to teach the course in the kind of interactive way that we want, and with the amount of grading help that we get in a larger size. We want to be able to do short answer questions instead of Scantrons. So that's why we keep the class in a manageable size. If you are an extension student, usually extension students also get spaces, but you need to hang on for 2 weeks and wait. I can't sign paperwork until we know there's spaces. If you are an extension student, I would appreciate it if you'd come up after class and bring me and write down your email address so I can keep you in the loop about course announcements before you enroll. Any questions? All right, now the last thing has to do with the podcasts. In previous years, I've used UCI Replay to record my voice and pictures that I'm projecting. This year, the course is being filmed. You'll see a fellow in the back who is recording this, and the goal is that eventually I might be able to use the lectures in a more flipped course way to have more time to discuss in class, and also potentially offer components of the course in a distance learning online format. So we're taping the class this year, and Sean back there is going to provide me with videos that will be posted on a private YouTube link, and those I will provide to you as opposed to a UCI Replay link. And he'll be able to capture me, the screen, and everything going on with the pointer as we give this. And I have a mic here which he's recording. Seems like it slid down a little bit, so I'll bring it back up. If there are any problems with the recordings or accessing them, we'll work on that. But they've done this a lot with other courses, and so we're going to try that this year. Okay, is it too dark? I think 75 might be better. Is that better? Or do you like to have it lower to see the screen? Let me know. Okay, let me tell you about what you're going to learn in this course before we jump into the actual content. You'll learn how the immune system functions to protect the host, meaning the person or the animal, and in some cases to cause damage to the host. And why is this exciting? It's because I think that this is a really fun course, an exciting course, because really you can apply all that you've learned from more basic courses in genetics, molecular biology, cell biology, to a complex biological system that affects our lives every day. We've all been sick. We all know people who are sick, whose immune systems are not fighting off viruses and bacteria or are causing problems. So understanding how this works is a really exciting process. And I would recommend to enjoy this course and to do better in this course, try to get beyond what do I need to know on the exam and try to think about what can I learn in this class about how life really works. Most of the students who stick with this class find it to be one of the most interesting courses they take, even though they usually say it's one of the most challenging. So again, there's a lot of material and some very complex concepts. I will go fairly quickly through much of the material, which is written down carefully in the notes for you to review later. But from experience, I know the areas that the concepts get difficult, and then I'll slow down. Nevertheless, at any point, if there's something that you don't understand, I would welcome questions, and I try to break during the class to answer questions at various points to wake people up and to sort of break the monotony. Also, not in the first lecture, but in later lectures, I always take a 1 to 2 minute break to just leave the lecture, drop the pencils, and talk about something topical and in the news or in our lives that relates to the lecture material. So that's our little break from the class that we'll have, to try to wake people up and put this more in the context of something more interesting. Now, I recommend that you read Chapter 1 as soon as possible. When I was in college, I would often wait weeks to start studying and reading, and I do not recommend you do that in this class. You should read the notes and the lecture and start reading Chapter 1. If you do read the book, you should start reading that right away. Okay. Here's some pictures of pathogens just to get us started. And we all know that the immune system protects us from pathogens, and we'll talk more about what pathogens are in a minute, but the immune system also protects us from cancer. And one way we know that is that HIV patients whose immune systems aren't working very well tend to have many cancers appear that don't appear in otherwise healthy people. So in this course, you're going to learn how the human immune system has evolved mechanisms to recognize a nearly limitless array of foreign invaders, some of who are shown here, as well as subtle differences between cancer cells and normal cells. You'll learn how the immune system eliminates the dangerous invaders in cancer cells. But keep in mind, as we move along, that many people still die of infectious diseases and cancer. So the immune system doesn't work perfectly, and our task as immunologists is to bring in other human advantages like intelligence and creativity to bolster the remarkable immune system we already have. And it's worth keeping in mind that really the immune system doesn't work as well as many other organ systems. In the absence of vaccines and antibiotics and all the other things we do to help us survive in this world of microbes, a large fraction of people die before they even make it to adulthood. That was the history of humanity. Death from infection on the battlefield, death of children and young adults. Whereas it's rare that somebody's born with a heart that doesn't work, or lungs that don't work, or kidneys that don't function. Those things happen, but they're very rare. It's almost anybody can die of an infectious disease if they don't have the right antibiotics handy or they didn't have the right vaccines. So really, our immune systems are playing catch up with these diverse microbes, and it's amazing that they do a job as well as they do, but we'd like to help them do better. It's also important to remember that the immune system is not always protective. The immune system can cause severe inflammation if the response is too strong or it's not turned off properly. You can get tissue damage, like all the scarring that you get from the acne in response to innocuous bacteria in your skin. Or you can even get death from septic shock or anaphylactic shock. So the immune system isn't always your friend. It's also responsible for allergy. I have allergies, many of you have allergies. And these, again, allergies are immune responses against harmless substances that really don't pose any danger to us. Yet we're trying to sneeze them out and cough them out, and other immune reactions that are really dangerous and can lead to death. From life-threatening anaphylactic shock, for example, to some peanut allergies and so forth. Then you have autoimmunity. If the response occurs against one's own tissue, then you get autoimmune disease. And this is really a very common set of diseases. A lot of people in the community don't realize it, but autoimmune diseases include multiple sclerosis, juvenile diabetes, rheumatoid arthritis, and lupus. And I'm sure many of you know people with these diseases and others, like Crohn's disease. So improper activation of the immune system against your own tissue is very, very common. I had a student in the class a few years ago who had juvenile rheumatoid arthritis. It's not just a disease of elderly people. And lastly, the immune system limits our ability to transplant organs because it wants to reject organs from somebody else. So at the same time we're trying to boost immune responses to these bugs, we're trying to limit immune responses to our own bodies. So I'm going to try to keep the arrow on here. All right, so a pathogen is defined as any organism that has potential to cause disease to the host. It's usually a microorganism or a parasite. Here's some pictures, and figure 1.4 comes in 3 parts, and it just is a long list of the different types of human pathogens. Now, I don't expect you to memorize this. This is just for your interest to, why are we sitting here talking about immunology? Because there's a lot of dangerous bugs out there, and these are some of them. Many of them you've heard of, some of them you haven't. Some of them cause diseases you were aware of but didn't realize that they had a microorganism involved. One thing you should do, especially if you haven't taken the microbiology course yet, is to review bacteria versus viruses versus fungi versus protozoa and general aspects of their life cycle. You need to understand viruses as intracellular parasites that replicate in cells, and bacteria as either extracellular or intracellular. Those sorts of distinctions of pathogen life cycles will become important as we talk about how the immune system deals with them. Now, some of these pathogens are called opportunistic pathogens because they only cause disease in hosts that are immunologically weakened or compromised in some way, like HIV-infected people, or patients undergoing immunosuppressive therapy after organ transplantation or cancer treatment. And then there are many, many microorganisms that are not pathogens, even though they coexist in our body. They're the normal flora of the gut, on the skin, and in some cases they actually help you. And I'm sure you've all heard about helpful bacteria and microbiome and probiotic therapies and so forth. And the term for normal gut flora is commensal species. In fact, we have more non-human cells in our body than human cells because bacteria are so small. A lot of them don't add up to a lot of space, but we have more of them than we have of ourselves. Now, even among pathogens, most of them don't kill the host. It's actually a better strategy to survive, to allow the host to survive and to transmit the disease by being really sick and coughing and sneezing or having diarrhea and then recovering, so that the person doesn't die right away and they infect as many people as possible. And that gives the immune system a chance to deal with it and allow recovery. Even severe diseases like influenza don't usually kill the patient, as the immune system eventually eliminates the virus. Now, all of these pathogens have molecules on their surface or inside of them that are different than our own molecules, and those are called antigens. An antigen really is defined as any substance that can induce an immune response. Okay, so before I get into the next slides, I just want to talk generally about some of the body's defenses and give you some definitions. In this course, we're going to talk about innate immunity versus adaptive immunity. Innate immunity is defined as mechanisms that recognize and control or eliminate pathogens without requiring any prior exposure to that pathogen. It's like a first line of defense. It's pre-existing in the host, and it's relatively nonspecific. You've probably heard about this if you've taken microbiology and virology. Just to remind you, that's innate immunity. Adaptive immunity are mechanisms that are not pre-existing. They develop over time after exposure to a pathogen. And they're mediated mainly by cells called lymphocytes. And two main classes of lymphocytes, T cells and B cells. And also, adaptive immunity is antigen specific. And finally, it provides memory. It provides lifelong immunity to reinfection. That's just the introduction. We'll come back at the end of this lecture to a little more details about innate and adaptive immunity, and then, of course, detailed lectures on each of them later on. So some definitions. I want to start with the term leukocyte is from Greek words that mean white cell. So leukocyte is a white blood cell. And that includes just about all the cells that we're going to talk about in this course are generally termed leukocytes. Within that group, the lymphocytes are a subtype that includes the T cells and B cells, as well as another cell type called natural killer cells, or NK cells. And while they're lymphocytes, NK cells are really more part of the innate immune system. And we'll see how that works later on. An antibody is a protein that binds antigens and is produced by B cells. Antibodies found in the plasma and other body fluids. What is plasma? Plasma is the liquid component in the blood. If you can imagine blood just removing all the cells from it, what's left is plasma. And plasma is constantly leaking out of the blood vessels into tissues where it mixes with other fluids that are in those tissues outside the cells. Together, this material is called lymph. You've heard of lymph nodes, I'm sure. Lymph is this material that is partly composed of blood plasma and other secretion and everything that's existing inside the tissues. Now, how do you obtain plasma from a person or from an animal? You can do it by removing the blood in a way that prevents clotting, and then centrifuging to remove all of the cells. So it's basically doing an experiment similar to what I just imagined, which is removing all the cells. But in order to do that, you have to take them out of the body and spin down the cells, but you have to do it in a way that prevents clotting. Once the blood clots, things change. And if you let the blood clot first, before you do the centrifugation, now the resulting liquid is called serum. Why is plasma different than serum? During the blood clotting process, many of the proteins in plasma get broken down and changed as part of the clotting process. In addition, platelets, which are involved in blood clotting, secrete a bunch of other proteins that are involved in wound healing, for example. And so the protein content of the blood changes during the process of clotting. And depending on what analysis you're going to do for a clinical analysis, or if you're going to use the liquid for some biological or biomedical application in the lab, you might use plasma, you might use serum. But I just wanted you to be aware that they're actually two different things. They're both a liquid component of the blood, but plasma is just really is the liquid component of the blood. The serum is the liquid after clotting has occurred. And you'll run into both terms as you read through the text, and I wanted you to be aware of the difference. Any questions about plasma versus serum? So now to return to innate immunity. Here is a picture of many of the surfaces that are places that pathogens could gain entry. You can breathe them in, you can eat them, they can try to get in through your skin. Once you breathe them in, they'll try to get across your lungs. They can come in through the genitourinary tract. If they make it past the stomach, they can take up residence in the intestines. So all these places are areas that pathogens, as well as symbiotic organisms, can live. And innate immunity is often concentrated in those areas. In fact, the anatomical barriers themselves are considered part of innate immunity. Skin is part of your innate immune system by preventing access of pathogens to your body all the time. In figure 1.6, you can see some of the other properties of the innate immune system and mechanical properties. For example, the mucus and cilia that we have in our lungs, trachea, esophagus, etc. This traps a lot of the microbes, and it gets them out as you cough, and helps you to cough them out. So then you have these epithelial cells that are joined together by tight junctions in your skin, in all those surfaces that I mentioned over here. Epithelial cells is a type of cell that has tight junctions to really serve as a barrier to the access of any potential pathogen that's trying to get across. So I mentioned earlier battlefield wounds. If you watch a movie about some battle that took place in 1500 or 1700, they always show people getting getting impaled by a sword or getting a head chopped off or something like that. But far more common was, some sort of deep wound or a gunshot wound to the stomach where the person didn't die right away. The reason that most people died on the battlefield until the advent of antibiotics was through infection. So they would linger for a few days and then get these horrible infections and eventually die. And that's because the barrier function of the skin was broken. So it's a really powerful component of the innate immune system, our skin. It's what you avoid having broken when you're in battle, and as they would clean up the battlefield afterwards, a lot of those people were still alive, and they would just go back to the medical tents and die a few days later. Okay, so going back to this slide here, some other aspects of the innate immune system that are sort of non-specific, it's hard to even think of them as an organ or anything like that, is that the low pH, the acidity of the stomach, takes care of a huge fraction of any pathogens that we take in through our food and drink. There's also proteases like lysozyme, which is present in our nasal secretions, it's present in tears, it's present in saliva, and lysozyme breaks down the cell walls of bacteria. And then there are also a whole class of peptides, short peptides known as defensins, which are secreted by epithelial cells all over the place, and they can poke holes in the membranes of bacteria. Now, not shown on this slide, but something we'll talk about is a process known as phagocytosis, in which cells internalize and break down foreign cells and macromolecules. But we will talk about this process known as inflammation. Inflammation is defined as tissue damage and leakage of vascular fluids in cells in response to infection. And it's all initiated to try to help fight the infection, but of course, if there's too much inflammation, that can cause damage. The innate immune system plays a critical role in the inflammatory process by secretion of proteins known as cytokines. And among cytokines, there's a subset called chemokines, which actually attract other cells and lead to permeability of the membrane. So here we go. In healthy skin, there's these effector cells, which is a general term we'll just use now to describe cells of the innate immune system. Here's the capillary, or the blood vessel, with some white blood cells, some red blood cells. And here's the epithelial layer or the endothelial layer of the capillary and epithelial skin. You fall down, you get some dirt and some bacteria get in. The effector cell of the innate immune system takes up the bacteria, releases cytokines. This leads to openings in the endothelial wall, allowing fluid plasma to leak in, allowing cells, white blood cells, to squeeze across. And then these additional cells, through the process of phagocytosis, taking up the bacteria, can eliminate any remaining pathogens. So the 4 cardinal signs of inflammation are heat, pain, redness, and swelling. And for those of you going to medical school, you don't need to write this down, but you may hear the Latin terms for these, which are calor, dolor, rubor, and tumor. Again, heat, pain, redness, and swelling. And the reason you have heat is because of the increased blood flow. Normally, the surface of your skin is more closer to the surface of the temperature of the air. Your blood's at body temperature. As you get more blood in that area, it warms up. It also causes redness because you have more blood there. The pain is because of pressure that's exerted on your nerve endings by the increased fluid flow. And then the swelling, of course, is because of the increased presence of blood plasma and cells in that area. So we're going to learn a lot in this class about cytokines, which are small proteins that direct other cells and tissues what to do. And among the cytokines, a subgroup called chemokines, which are specifically involved in attracting or repelling other cells to get them to move in different directions. The pointer's not staying, I don't know why. Visible. All right. So, any questions about inflammation?
So it's the cytokines that sort of make the membrane more permeable, or the cell capillary?
I think it's partly, it's the cytokines in general, but among the cytokines, the chemokines are particularly important. Actually, I would say both chemokines and other types of cytokines that are not involved in attracting cells are both acting on the cells of the blood vessel to make them more permeable. And we'll come back to some of the names of those proteins in the later lecture. Now, let's return to the question of how the innate immune system recognizes the danger, because before you do anything about it, you need to recognize that there's something wrong. So there are 2 phases of the innate immune response, the recognition phase and the destruction phase, which is also known as effector mechanisms. And as an example, I'll use a part of the innate immune system known as the complement system. Complement system are a group of proteins in plasma that bind to pathogen surfaces and mark them as dangerous. So here's a bacteria, here's complement proteins. They have a way of recognizing bacteria, which we'll talk about later, and forming a covalent bond and covering the surface of the bacteria with bits of these complement proteins. Once you have this coating of the bacteria, there are receptors on the surface of innate immune cells that can recognize the complement fragment, and that leads to binding and internalization of the complement-coated pathogen and phagocytosis and destruction of this bacteria inside this cell. Now, cells of the innate immune system can also directly recognize pathogen surfaces that have common structures that do not vary from one bacteria to another. So we call these pathogen-associated molecular patterns, or PAMPs. An example would be gram-negative bacteria have a structure called lipopolysaccharide. Gram-positive bacteria have other cell wall structures, and all bacteria of those classes have those invariant conserved structures, and we have evolved receptors that basically stick to them, and when that recognition event happens, that tells the cell to spring into action to do phagocytosis or release cytokines, or whatever their job is. So again, one way is through these complement proteins that are in plasma and lymph and extracellular fluids, which tag the bacteria or pathogen for recognition. Another is directly through, not shown on this slide, pathogen PAMP recognition receptors, which can directly recognize that there's a pathogen-associated molecular pattern. Okay, we're moving along pretty well. Now I want to give you a brief introduction to adaptive immunity. When I was a graduate student in the early 90s studying immunology, this is all anybody wanted to talk about. It's really fascinating, and people didn't appreciate the importance of the innate immune system. And now the pendulum has swung back a little bit so that a fraction of the immunology research community is focused on the innate immune system and how we can harness that to improve vaccines or to reduce inflammation or deal with certain types of infections and autoimmune disease. Nevertheless, the adaptive immune system remains fascinating, and it will be really the focus of much of the first half of the class and much of the material that I'm going to present. So I'll do a little bit of a preview today and be prepared for a lot more details later on. Adaptive immunity, as I said, is antigen specific, and that means it can distinguish one strain of bacteria or a virus from another. Where I said that all gram-negative bacteria have the same structure that is recognized by an innate immune receptor, the adaptive immune system, the T cells and the B cells, can distinguish one strain of E. coli from another. That just can't be done by the innate immune system, but only by the adaptive immune system. It can distinguish closely related viruses and mount a response against one and not the other. But in order to do this, it requires a delayed period of 4 to 10 days. And we'll see why that is in a moment. Adaptive immunity provides this long-lasting immunological memory so that a second encounter with the antigen produces a much stronger response and also a much faster response. This is the basis for vaccines. This is why vaccines work. It's because it's basically educating your body and giving it a memory of a pathogen so that if you ever are exposed to the real thing, you have a fast and strong response and you never feel sick. Again, the memory is specific so that a vaccine for one virus doesn't protect you from another. A vaccine from one bacterial toxin doesn't protect you from another. This memory and any aspect of adaptive immunity is mediated by lymphocytes, T cells and B cells, and in the case of B cells, it's mainly mediated by the antibodies that they secrete. T cells have a very diverse set of jobs that they do. B cells, their main job is to secrete antibodies. It's also important to realize that innate immunity and adaptive immunity talk to each other. We'll see how innate immunity signals the adaptive immune system to get into gear, but it's also true that adaptive immunity, the effector mechanisms, the adaptive immune system helps to increase the efficacy of the innate immune system by focusing its effective elements at the site of invasion and enhancing responses such as phagocytosis. So they really work hand in hand. The innate immune system is the first line of defense. It also signals the adaptive immune system. And once the adaptive immune system has gotten into gear, it will help the innate immune system do its job better and in the right place. So why is it that you can recognize these subtle differences in using an adaptive immune system, and why does it take so long for it to develop? And that's illustrated mainly on figure 110, which talks about clonal selection. But first I want to define a couple of terms. First is proliferation. You probably understand this, but it basically refers to cell division that leads to expansion of a single parent cell into a population of clonal daughter cells. This happens during embryonic development. It happens in many tissues in your body that need to regenerate themselves. But many tissues in the body are not regenerating and not proliferating. And lymphocytes, the vast majority of them are not proliferating either, but in response to recognizing the antigen, they proliferate like crazy. And you get a huge population of clonal daughter cells of the original parental clone. Differentiation refers to a change in the phenotype and function of the cell. It's often irreversible. So what we'll see is that during the process by which T and B cells proliferate, they also differentiate and change their functions and become related cells, but with new or different functions. Now, the T and B cells, the reason that they're so specific is because each one has a single antigen receptor, and the sequence is unique to that cell. And that this kind of sequence diversity is unprecedented in biology, and it's generated through a process of DNA recombination that involves the random cutting and splicing and modification of small gene segments, and we'll cover this in some of the later lectures. The outcome of all this is that each lymphocyte has a different antigen specificity, and you can sort of see that illustrated in figure 110. This just shows 7 different cells, each with a different color, and each with a slightly different structure of its antigen receptor pointing here. Remember, there's thousands of copies of these receptors on each cell, but the structure of the receptor on one cell is slightly different from the structure of the receptor on another cell. It's illustrated in 2 dimensions here as little bumps or caves or different kind of subtle features to the surface of the antigen recognition molecule. And that differs from the receptors of the innate immune system. If you look at 10 effector cells, and an example is a macrophage that we'll learn a lot about, or a neutrophil, they have 10 or 15 different pattern recognition receptors that are useful for recognizing gram-positive, gram-negative, fungi, viruses, etc. But the sequence of those receptors is the same on all the macrophages and all the neutrophils. In contrast, the sequence and the fine structure of these antigen receptors is different on every T cell from each other and on every B cell from each other. And you have billions of these T cells and billions of these B cells, and when you get infected with any one pathogen, only a very small number of these will actually have a receptor that makes a good fit with the antigen that is presented or present on the surface of that pathogen. It's these clones that are selected to fight the invader. From the huge pool of potential T cells or B cells, only those clones whose antigen receptor fits well with the antigen are selected. The clonal selection refers to the proliferation of these antigen-specific clones that are going to be the ones that are useful to combat the infection. So that allows your immune system to be prepared to respond to an infinite array of antigens through this huge, diverse repertoire of cells. But each pathogen selects only a few cells that then make many, many copies of themselves. So here, a pathogen is illustrated as a little round antigen here, and it seems to fit nicely with this yellow cell. Of course, it wouldn't fit as nicely with these others. And that clone is selected and to proliferate and make many copies. Shown here are 5 copies, but typically there might be 100 or more daughter cells that are produced during this process of clonal selection. And that process takes time. It takes several days to make hundreds of copies of the original cell, and that's why adaptive immunity is slow. All the cells of the innate immune system are already there. They have the receptors already expressed. You have a lot of the cells in your tissues. They can respond immediately, but they're limited in how well they can differentiate one cell type, one pathogen from another. Here, innate immune system, adaptive immune system, you have billions of different specificities. 1 or 2 are selected to expand, and after several days, now you have your little army ready to fight the infection. During this process, the cells differentiate to acquire effector functions. For example, the resting B cell can't secrete antibodies. Only during the clonal expansion phase do they differentiate into a type of cell that can really secrete antibodies that are useful in the infection. And we'll discuss the functions of effector T cells and effector B cells in great detail later. Now, figure 1.9 compares some of the concepts I've just gone through in innate immunity versus adaptive immunity. Again, innate immunity responds rapidly in hours or even minutes, but it's a fixed response that doesn't differ between cells or even between individuals. And there's a limited number of specificities. We've evolved receptors that can recognize general features of different classes of pathogens, but there's a finite number of those available to us. And it doesn't really change much during the response. There's no alteration in the army that's being used to fight the battle. The difference in adaptive immunity is it's a very slow, it can take days to weeks. The response is variable. Only those that are useful for the infection are selected. And then you have a lot of these specificities, billions of different T cells and B cells. And over the course of the response, the immune response, the adaptive immune response improves. You get more and better cells with increased ability to detect and destroy the antigen. Eventually, though, they both converge on a similar set of effector mechanisms that actually destroy the pathogen. So we need to remember that eventually there's only a limited numbers of ways you can destroy a pathogen. Phagocytosis is one, direct lysis is another, and so we'll learn about the ways that we actually destroy the pathogens and how innate immunity and adaptive immunity control those effector mechanisms. So I said that when I was a graduate student, everybody seemed to be interested in adaptive immunity, but this graph really makes it clear that, if anything, innate immunity is more important. So if you do an experiment with an animal that lacks innate immunity, or a human who's had their bone marrow eliminated through radiation or chemotherapy, so they can't make any cells of the innate immune system or adaptive immune system, and then they get infected, the microorganisms just replicate super fast exponentially. There's no first line of defense. And if you genetically remove certain genes and cells of innate immunity in a mouse, even leaving adaptive immunity intact, this happens, the microorganisms will outpace any adaptive immune response. If you only are lacking adaptive immunity, compared to normal humans, the early phase is fine. You control the expansion of these microorganisms. But there's a point normally where the adaptive immune system kicks in to destroy the remaining cells. If you don't have adaptive immunity, those microorganisms will now slowly overwhelm you. So this would be an example of somebody with HIV. They're lacking certain types of T cells, but it might take months, years, or decades to reach the point where the immune system can no longer deal with infections. But somebody who's had radiation or has been a burn victim so that their skin is largely removed and the barrier to microorganisms is removed, they can die within hours or days from massive infection. And the last slide is just, the last two slides are just to illustrate the concept of immunological memory, and that the secondary response to the antigen is faster and stronger. Now, in this example, an animal or a human is getting two vaccines, one against antigen A and one against antigen B. And then you're measuring the antibodies in the serum on a log scale here that are specific either to vaccine A or vaccine B. And what you can see is the person's vaccinated just with A here. It takes a little less than a week to start measuring antibodies. This is the adaptive immune system lag phase. And then the antibody response goes up, and after a couple weeks it reaches a plateau and then slowly starts to go away as the antibodies are degraded. But remember, this is a log scale, so you still have quite a lot of antibodies left. Now here, the person is vaccinated with a mixture of A and B. And you can see that the secondary response has a very much shorter lag phase. It's almost immediate. And you get many logs of increase in antibodies. So it's faster and it's stronger. And then the plateau is much higher. So you may have had booster shots. You can imagine the booster shot is giving you a very large and rapid, huge increase in the amount of antibodies to whatever that antigen is. But in the same vaccine, the response to the component B is just the same as it was for A and the primary response. So just because your body's responding faster to antigen A doesn't make it respond better to vaccine B, because this is a primary response to vaccine B. At this point, the patient would have protective immunity to any pathogen that had A in it, but probably would need a booster shot to get a really strong protective immunity to B. And lastly, just to illustrate the power of vaccines, diphtheria is is a bacteria that produces a toxin that's very deadly. And in the first half of the century, it actually was very common. My father, who was born in 1923, nearly died of diphtheria toxin. The vaccine came in around World War II, and then within a decade, the frequency of diphtheria cases was almost reduced to 0. Polio was a disease that only became prominent in the middle of the 20th century. And you could see it became very common and was it could be life-threatening and disabling disease. And then the Jonas Salk's vaccine and then the Sabin vaccine came in, and you could see the dramatic effects they had to reduce the cases and eventually eliminate polio largely from our world. And interestingly enough, with measles, again, the vaccine came in, measles went away, but there's a disease that occurs 20 or 30 years later that is a neurological disease that you could see followed in the same course. It occurred in these people who had measles early on in the 20th century. Later on, they got this disease, and then there was a lag phase for after the vaccine eliminated the primary infection. Later on, you stopped getting people with this disease. So you're going to hear me advocate for vaccines a lot in this class, and we can refer to this figure. It just shows you the power of the adaptive immune system, immunological memory, and vaccines. Okay, I'll see you Monday. Last announcement, I put this in the email yesterday, but I will always post the lecture notes and slides ahead of time, yet I still think there's value in coming to class, asking questions, and hearing about the context. All right.